Okay, we can probably officially get started. Wenbin, can you record the presentation? So we will keep all the presentations recorded so that we can send out the links later on if you guys want to revisit. Can you all hear me all right? Okay, good. Welcome to the SEFARI 3D-RNA-seq
This meeting is being recorded.
workshop. And just a few things. First, I would like to acknowledge SEFARI for the funding to hosting this virtual workshop. And the workshop organizers include Wenbin Guo, Linda, me, and Ian. We are all from the Information and Computational Sciences from James Hudson Institute. So, yeah, just as you know, the meeting is recorded. So to minimize the background noise, we would encourage to mute your microphone during the presentations. But you're welcome to put your camera on to increase the interactions. So we will leave time for Q&A for each presentation. But as I said, we have a relatively small audience. So if you have any questions, feel free to interrupt and raise on the spot as well. And alternatively, if you feeling more comfortable, you can leave your questions in the chat box. And yeah, we can make summary of the Q&As in the chat box and make it available online after the workshop as well. And more importantly, we also have a Google group for the 3D-RNA-seq that can be used at any time, even after the workshop. If you have any questions, you can raise your question on the forum. So the rough schedule for today, we will have a welcome. And for the first part of this training, I will give a presentation regarding to the transcript quantification part. And then after that, Wenbin will give us a demo, basically to show you how to do that on the EU Galaxy, which is actually a free server you can use. And then after that, we will have a break. And then we will have another talk. Wenbin will present the details of 3D-RNA-seq. And then followed by another demo by Wenbin. It will show you using an example data set to show how that works in real time. And finally, we'll have Q&As and a wrap up. So just as I mentioned, we have one-to-one sessions time allocated for next Friday. So you have time. You will have time to play with 3D-RNA-seq. If you get stuck or get any problems, feel free to book one of those sessions. Please contact Linda for this. And we can walk you through that one-to-one. Because doing the training online is a bit difficult. If we have any question, we can only looking at one screen at a time. So that's why we think this could be a better way. And also, if there's any suggestions from anyone to improve the workshop, please let us know as well. So finally, the recording will be made available online after the workshop. And there's also several useful links. You can find more information about 3D-RNA-seq or a GitHub page, and you can find the user manual there. And actually, the last two, which I would think that's the most useful. So when we have made a very comprehensive tutorial and put it on YouTube, you basically can follow that tutorial. You can stop anytime to adjust it according to your speed. And then the Google group, as I mentioned earlier on, if you got any questions not covered by the video, you can raise it there. So just briefly, any quick questions before I proceed with the presentation?
Renshuan, the one-to-one section is on next Thursday, not Friday.
All right. Oh, sorry.
Yeah.
Okay, yeah, Thursday. Good. Okay, I'll get started with this part of the presentation then. So in the first part of the presentation, I will talk about accurate and fast quantification of transcriptomes using RNA-seq data. So the RNA-seq has been available for us to use for about 15 years now, since 2005. And it promised more diverse transcriptome compared to the early technologies of microarray. And it allows transcript level quantification with improved gene level quantification as well. And more importantly, I think the RNA-seq data allows the study of another layer of regulation, which is alternative splicing. So just for the people who are not familiar with alternative splicing, so here, this example that alternative splicing can selectively choose the exons that you can incorporate into your transcript. So from the same gene, you can have two transcripts actually constructed just by the alternative splicing regulation. So the challenges of RNA-seq data analysis actually comes from many levels. First, RNA-seq, what we get is actually the reads. So how can we derive those reads into the gene or transcript quantifications? And the second one will be differential expression. Because RNA-seq data follows a different distribution. For example, it's a negative binomial distribution. So that means we need new statistical models to carry out those statistical tests for differential expression. And finally, the alternative splicing analysis. Although, as I said, the RNA-seq data have the information there, but this analysis is largely ignored, mainly due to the poor transcripts and alternative splicing annotations. And there's significant amount of information that has been lost if you do not use it in your RNA-seq experiment. So first, I'm going to talk about a few issues with gene level inference. So this is one of the figures I quite like from the Nature Biotechnology paper. So if you want to infer the gene level quantifications, one of the ways is that you should get your transcript quantification first, so they can edit them up. But if you don't do your transcript level quantifications, there's only two models you can use to get gene level quantification. For example, the first one called the uni model. So this is hypothetically, this is two isoforms within one gene. So you have this red exon here and I have blue exon here. And the uni model, they basically take the unions of all the regions that this two isoform covers. So this is the uni model. And the intersection model means that you only take the overlapping parts of these two isoforms, like this. So what the result shows here that if you use the uni model or the intersection model, the fold change you estimated from the read counts are wrong. So this is basically the true expression is 0.3, but if you use a union count, you actually don't see any changes at all. But if you use the intersection model, the fold change will be estimated lower. So this is basically three scenarios. You can see that the union models and the intersection models all get the quantification wrong. So that's actually has been reached a consensus in the community that these two models are not correct. Even if you don't care about the transcript level analysis or the alternative splicing analysis, you're just interested in the gene level analysis, whether genes are differentially expressed, you should still get your transcript level estimates. So this is a tweet from Leo Pachter. So if you guys know him, he's developer of the Callisto, one of the top labs for RNA-seq computational methods. So he, in this Twitter, he mentioned he found it frustrating that feature count is still used to quantify RNA-seq data. So feature count is one of the methods using this union or intersection approach. So again, this paper published, union exome-based approach for RNA-seq gene quantification to be or not to be. Clearly not to be. And then there's also a recent paper shows that differential analysis of RNA-seq data, the transcript level estimates improve the gene level inferences as well. So, so we know that recent development of methods like SAM and the Callisto, they allow alignment-free transcript quantifications. So they have been the standard of the state-of-art methods in the field for a number of years. They are very fast compared to the mapped-based methods. They also save time and data storage. They are also one of the most accurate methods up there. But the issue is that they do require a transcript reference. So the transcript reference to an RNA-seq experiment is almost like a dictionary to a book. So for example, you want to find out which word has appeared in the Game of Thrones and which word has appeared how many times in that book. So, but unfortunately, the issue is that existing transcript reference is typically incomplete and poor quality, particularly for plant species. So for the best annotated plant species out there, which is Rhabdopsis, you can see that the TER10 or the Aeropoint 11, which is more recent kind of annotation, we know there's tens of thousands of alternative splicing events and transcripts are missing there. So if you look like that, it's an incomplete dictionary. But at the same time, right, so if you got your RNA-seq data, one other alternative is that you can map your RNA-seq data to the genome reference and assemble the transcripts by yourself. But the issue is that the current state of art of tools like cufflinks and string tie, etc., they misassemble, according to different literature, they misassemble, more than half of the transcripts. And at the same time, more than half of the transcripts actually in your sample, they are not assembled at all. So that's really a big issue. And the issues with this transcript annotation, including, for example, redundancy. You have the same transcript represented multiple times, and you have transcripts with false splice junctions, or they are just actually fragments of the transcript instead of complete transcript annotations. So we started this journey back in 2014. So we are one of the first few labs actually to study the impact of the transcript annotation to the transcript quantification. So I started this collaboration with Professor John Brown at University of Dundee and Professor Hugh Nemo in University of Glasgow on a very big set of RNA-seq data. So I just described the experiment briefly because we're going to use it in the experimental data set, exemplar data set for the 3D RNA-seq as well. So the plan was Arabidopsis plant was harvested at 20 degrees for every 3 hours for 24 hours. And then the plant was shifted to 4 degrees. And then the sample was carried on every 3 hours again for 24 hours to capture the transition response. And then another 24 hours sampling was carried out at day number 4 to capture the more acclimated conditions. So we have 26 time points altogether with 3 biological replicates for each of the time points. And in total, we have about 5 billion pairs freeze. So that's quite a big set of data. So the challenges at the time to analyze this is how can we accurately measure the gene expression and also alternate splicing, particularly alternate splicing. And this is also a time series RNA-seq data. So I visited one of the top computational biologists working in human alternate splicing at Eduardo Iris. So currently he's with AMBO in Australia. And using his advice, we decided to use sailfish salmon pipeline. So sailfish is early version of salmon, less accurate, but at the time it was still the forefront. So you can see the timeline over here. Because we know at the time the TER10 was incomplete. So we incorporate a lot of new transcripts discovered from this Marcus et al. So we want to make the database more comprehensive. And at the same time, we generated high resolution RT-PCR alternate splicing data that actually allows us to validate when we make changes to the transcript annotation, how that can improve or affect the performance of quantification. So we actually went through several iterations of optimizing the assembly merge pipeline. And we identified issues, for example, obviously, if you got missing transcripts, your quantifications will not be accurate. And also issues including redundancy, as I mentioned earlier, and also fragments. You can see the complete transcript is like that, and you have little bits of fragment of that transcript. But there's another issue that we do find some fragments here, but it contains some novel events. So removing those fragments will cause us to lose those novel information. So what we decided to do is that because we have this huge amount of RNA-seq data, so we decided to assemble those transcripts ourselves. And to overcome the issues with missing transcripts, we contact our collaborator in Vienna, Andrea Bata and Maria. We put together a range of a large set of RNA-seq data with different tissues and different stress conditions to capture alternate splicing as much as possible. And at the same time, we applied a number of quality filters to remove those misassembled transcripts. And I can show you here, just here, the impact of one of the filters we developed. So the transcripts, if we remove transcripts with poorly supported splice junctions, that removes about half of the transcripts put together by cufflinks. And that's 30% of the transcripts put together by a string tie. And then at that time, Airport 11 was published. So we decided to incorporate Airport 11 to make the annotation more complete. Then we come up with another issues because Airport 11 tends to have very long UTR regions. And we identify that the variations caused by misannotation at the UTR regions actually affect the quantification results as well. And this was later confirmed in a different study published by Mark Robinson's group, which is also one of the top statistical group for RNA-seq data. So they identified the same problem in human data as well. So quantification is strongly affected by the misannotation, the 3' UTR. So what we come up with is so-called ATRTD quasi. So that's quantification of alternatively spliced isoforms. So we padded the transcripts with the genomic sequence so that all the genes start and end at the same genomic location. So that was designed specifically for quantifying the transcript isoforms. So you can see one validation here for this gene. We have an alternative splicing event over here. We calculate the ratios between this alternative splicing event versus the canonical event ratio. So the high resolution RT-PCR shows that the ratio is only about 3%. But when we using the kind of unpadded version, we can see that the ratio is actually 30%. It's tenfold more. But then after we did the padding, the ratio was corrected back to around the same order, 2-3%. And then we also look at the splice junction rate counts, and that ratio is also about 2%. So it just shows that the RTD quasi overcomes the effect of this misannotation. And as I mentioned early on, we have this high-resolution RT-PCR panel developed. So we generated this plot basically using the quantifications, using the high-resolution RT-PCR data, and also the RNA-seq data with different transcript annotations. You can see that this is ATRTD2, and this is ATRTD2 trimmed. And this is padded, ATRTD is padded. And this is the airport 11, which is the community annotation at the time. Altogether, we actually have 762 data points covering 127 alternative splicing events from 62 genes. And in terms of correlation, we can see the RTD2 quasi achieves Spearman correlation of 0.8 and 0.9 for Spearman correlation. And you can see for airport 11 is far lower with 0.5 for Spearman and 0.37 for Pearson correlation. And even just from scatter plot, you can see the RT2 quasi, the consistencies between the two quantifications is much similar. So all these details are published in NAR in 2007, if you want to have more details about the methodology. And of course, after that, we also see a number of publications confirming the issues we find with transcript annotations. For example, this one, as I mentioned, in 2009, early on. And there's also this paper in Scientific Report. They also find the assembly completeness, also the overlap, the redundancies, for example, here affects the quantification as well. So here is a similar information we find in another paper just published recently. So the pipeline we developed has been translated into Barley. So the BART one was produced by my colleagues, including Paulo, Linda, Misha, and Craig. It's published in 2019 BMC Genomics. And this is, we're also working on a potato RTD as well, which was done by Juan Carlos, our PhD student. And a visitor from Malaysia oil palm panel, oil palm board, also came over and he constructed this RTD for oil palm. And we also got a BBSRC grant funded from last year. So we are going to put all these methodologies into a pipeline, allow people to construct this transcript annotation for any species. So in addition to the short pipeline I just described early on, and we will incorporate the long read pipeline as well in this project. So we're expecting to have this pipeline ready probably in 6 months or a year's time. So if you have need to do that, get in touch and we can probably talk about it. So finally, I would like to acknowledge all the people involved in this. Professor John Brown, a long-term collaborator, and also Wenbin did a lot of this work on the computational side, and Chris and Nicoletta was a postdoc working in the wet lab generating data. And also Hugh and Alan, our collaborators at the University of Glasgow. And that's it for my part. Time for questions. If not, we can always discuss that later on when you want to talk more about it.
Have you got a list of those papers that you suggested?
Sorry?
Have you got a list of the papers that you listed that were written following your research?
This part. Is this paper or which
You
paper?
had a slide with some papers about RNA sequencing. Could you just put that slide back up so I could just make a few notes of those, please?
All right, okay, yeah, no problem at all.
Thank you. Thank you.
Is this the slide?
Yeah, that's perfect. Thank you.
Yeah. So I think I need to update for the last one, detect anomalies in RNA-seq quantification. I think it's already published somewhere. So if you search the name and the authors, you should be able to find the final version.
Okay, thank you.
Yeah. And also this one is our paper. Okay, so if no more questions, I will. Oh, comment. Yeah.
Sorry. Yes. So
Yes, please.
for BART, there is another more recent version, right? For BART.
Yes, we do, but we're still working on it. It's not published yet.
Okay.
So if you need to do it, use it. Contact us. I think we can give you access. Yeah.
Thank you.
Yeah. So if no more questions, sorry, go ahead.
I have a question. I have a question about this splicing variance. So primarily you count as if it come from same genome region you count as same, or you kind of distinguish isoforms in the result?
We can distinguish isoforms in the result.
Okay, so the result, so for example, if some isoform is missing in one condition, you can detect it.
Not exactly detect. So, for example, our RT-PCR data sometimes will identify products that are not covered by the annotation, so we will know that transcript is missing. So we construct this RTD. We want it to be more comprehensive, but it's also a moving feast. So I think, nothing is ever complete. As to say, you just want to, as complete, comprehensive as possible. Obviously, the RTD2 is way more diverse than the, for example, the current annotation pair 10 or airport 11.
Okay.
Yeah.
Okay.
Yeah.
Thank you.
And later on, if you will see the power of this transcript level analysis in 3D RNA-seq analysis.
Okay.
Yeah.
Thank you.
Yeah. Okay, thank you.
I have a question about BART version 1. The BART version 1 is based on the which version of the genome, barley genome? I think it might be version 1 of barley genome, I guess.
I think what I can say is definitely not the latest version, because the latest version published in 2019, I believe.
Yeah.
So,
I mean, are you going to update this with the latest version? Is there any plan?
yes, we do. Just as Carmen said, we actually already developed a version of BART 2.
Okay. Okay.
So BART 2 uses the latest genome reference.
Okay.
And currently we're writing up the manuscript. We will make it available as soon as we can publish it. Or I think if you can contact Robbie, ask about it. If you need to use it, we could give you access.
Okay. Yeah.
Yeah. Good. If no more questions, I will stop sharing and hand it over to Wenbin for a demo of this transcript quantification.
Yeah. Yeah, so I'm going to do the demo of transcript quantification with Galaxy. So everyone can see my slides, right?
Yes. Yeah.
Oh, great. Yeah. So I'm going to do the demo. So the tool we are going to use is Salmon, and we can also use Crystal for the quantification. And then you can find these tools in the Galaxy web interface. Then with this quantification, in the next step, we can do the 3D analysis. So I'm going to use a small dataset for the demo. The dataset is a subset of examples from the Arabidopsis study in response to the code. I'll also use the ATRTD2, the Arabidopsis reference transcriptome, for the quantification. So these tables show that samples are picked from the dataset. So I take the 10 point T2 from different day, and then each with 3 biological replicates. Then because the datasets, the RNA-seq reads are paired end reads. So here we can see we have the sequence file of read 1 and sequence file of read 2, and they're paired. And then in the Salmon quantification method, it will count the number of these RNA-seq reads that could be mapped to the input transcripts and then do the quantification. So here we are going to use a Galaxy Europe. So here's a link. And in order to upload big data sets, you need register a Galaxy account. So let's go to the Galaxy web page. So here is how the web page look like. You will have 3 parts. For the first part, you will have the tool list. Then in this part, you just type a name of the tool. For example, we want to use salmon, then we just type the salmon here, so we will get the tool salmon. Then in the second part, in the middle part, you will see the interface of the tools. So you have different boxes, you can import the data or you can set the parameters. The third part is the history part. You can see the panel here, it has a list of all the performance you did for the analysis, like the uploaded data sets, and then your performance of the analysis. So to do the analysis, we need the import data sets. So here I just show you how the data look like. So here we have the ATRTD2 transcript sequence FASTA file, and then we have the short RNA-seq reads FASTQ file. So to do the analysis, we can create a new history in the third part. For example, here we just click the plus button, and then there's a new history appear. Here we can give a name like 3D RNA-seq. And then we need to upload the datasets for the analysis. So here we can upload here, or in the first part, we have this up arrow. We click it, within this pop-up window, we can choose local files. So the dataset is here. So for example, we select the ATRTD2, and then it will showing here, we start the upload. Then you'll be adding to the history. For big data, it may take a while to get uploaded. So let's just wait for a while. So you can see there's a new item showing in the history. So it's uploading. Then we can also click the upload button and then choose local files. And then we upload all the samples, the RNA-seq examples. So we can start, and then the things will be uploaded. But it will take a while for that. So I already uploaded the dataset. So if you go to users, I just go to my history record. So I have the datasets being uploaded. So once you finish the upload, you can see your ATRTD2 is uploaded here, and all your RNA-seq reads files uploaded here. So we can click the I button to visualize the datasets. So we can see for the ATRTD2, we have the transcript name, we have the gene name, we have the sequence of the transcripts. And then if we visualize the short reads files, we can see we have read 1, read 2, we have so many reads here, and then it's a short sequence of the reads. So then we can do the quantification to count or the number of these reads being aligned to the transcript sequence. So we are going to use the salmon quantification tool. We click here. So in the tool, we have many boxes to select the parameters and select the import files. So for example, select the salmon quantification mode. So it's RNA-seq reads. Then we use the reference transcript datasets from the history because the ATRTD2 is already in the history. So we just select the ATRTD2. Then we can set the camera runs, and then the data import is library is paired end. So we can select the paired end. Then it's a bit weird. So this seems it seems something wrong here. Okay, the data has been deleted. So I'm going to use another set. Yeah, I always prepare backups, so don't worry about it. So here we select the ATRTD to the reference, and then we select the paired end. So because all the paired datasets has been uploaded, for example, we can select sample 1, read 1 for the mate pair 1, example 1, the read 2 for the mate pair 2, and then we can also set the read type. So it's because the dataset is unstranded, so we just select the not strand. Then you also have other parameters for that. But if you want to know more details of that, you could go to the Salmon quantification user manuals, and then to get more details how to choose these parameters. Then we can, in the end, we can click the button execute. Then there will be a new job being running here. So you will do the calculation of this sample to the ATRTD2 reference. But the problem is, you can see here we only do the analysis for one samples, but in the input datasets we have six samples. So how to do the quantification for all the samples? Then we need build an object called data collection of the pairs. So what we can do is, here we have this tick box. We click it, then you will see a box just appear here. Then we select all the RNA-seq reads. So we don't select the transcriptome, we only select the reads. Then for all selected, we can build a list of data pairs for that. So from the pop-up window, we can see the import RNA-seq reads will automatically paired according to their identical patterns between these two file names. But if you want to use other ways to pair it, we can also do unpair. Then we just type the patterns we want to remove from these file names. For example, we want to remove the 1.fastq.gz and then fastq. Then we just auto pair again. Then you will see the labels of the paired files are just changed. Then we can give a name to the file. For example, it's read pair. Then we can create the list. So in the history, we can see there's a read pair here. And if we click in, we can see all the examples are showing here. So if we click in again, we can see it's forward and reversed. So the RNA-seq reads are paired here. Then let's back to the Salmon quantification panel. So we have the reads, we have the ATRTD2 as a reference, and then from here in the library made paired, we select paired end data collections. Then we can import this read pair, and then we can set different parameters, and then in the end, we just to click the button to launch the job. Then in this window, we can see the analysis have been performed on all these paired samples. So this is how the Salmon quantification do the job. And then also in the history, we can see there's a job is running. So all the samples are being quantified, but it will take a while. But then I'm going to show something about the crystal. So for the crystal, we can also search the tool here, and for the crystal quantification, And then we also have this interface, just similar performance. We select the transcriptome from reference, then we select the fast ATRTD2 as a reference. From the single end or paired reads, we select paired, and then collection or individual data sets, because it's a pairs of list of the collections. So we have this. Then we can set different parameters. You can go to the user manual for more details for the parameter settings, and then we run to the job. Then for the crystal, it's also doing the quantification. For the crystal, you have two formats of output. One is tabular format, and one is the HDF5 format. So for the 3D analysis, we are going to use a tabular. I will go to give more details about that. So you can see the job for crystal is also running, and the salmon quantification, the job is already done. So if we click, it's running. Just don't worry. Then I go to my history record. I have a job finished here. So you can see once the jobs are done, it will be turned to green. So if we look into this output, so actually the output is a table. So this table lists the transcript names, the transcript length, then TPM values, and then the number of reads. So also the same for crystal. If we click in as different examples, then the table showing the quantification results, the color names are slightly different, but the same information is the transcripts, the length, and the recount and the TPM values. So for the 3D analysis, we need download these datasets. So if we click into the job of salmon quantification, then here we have a button to download the collections. So we just click it and then download it. So in the download, we will see a downloaded file like this. So if we click in, then the quantification of all your samples are here. And then we will use this result for the quantification in the next step. So that's the demo for the quantification. So if you want to So if you want to have more details, you can go to the user manual. So in my GitHub page, then in the user manual part, you will get the transcripts quantification, use Galaxy, the user manual. Then you can also go to the YouTube videos. So for the first part, I just show you how to use the Galaxy for the quantification. So don't worry if you didn't follow me. So you can go to the YouTube video for more details.
It's very difficult to do that. But I would emphasize that for the handouts you get, you actually also have a menu, one of the files called transcript quantification using Galaxy. Have you got that file? So that's actually very detailed information. So Wenbin basically put every step in that menu. If you just follow that menu, it should work out. It's also quite graphic. So Wenbin put screenshots of the Galaxy network, the interface, and basically annotate every column there. If I can show you, here. So you should all receive a number of files from us. So one of them is transcript quantification using Galaxy. Do you have it? Okay, yeah. So you can see Wenbin actually put all the details there. So if you want to play with it, you can use the download link to download the data here. So if I click, it will take me to the Dropbox where that data is. So you just need to download the data from here, the Dropbox, and then start. So we may have put screenshots here and all the annotations for different tools where you should look at things, how to upload data to Galaxy, etc. So yeah, I think with this menu and also the YouTube video, that should allow you to walk through that Galaxy pipeline. If you have troubles, don't hesitate to get back to us. Particularly, we have slots available for next week. Okay, yeah, that's all I want to say. Any other questions from people so far? In that case, we should take a break.
There's a question in the chat box from Carmen. What's the difference is choosing SAM or crystal? So in the very old version, there's a blog like saying that crystal and SAM, their similarity are very close. Is that the result is the consensus is about 99.99999% similarity. But in the latest version, seems they incorporate more like functions, so the difference could be a little bit higher, but still they are very similar. And then different labs, they just prefer their own tools for analysis. And then we also apply the 3D analysis for different data sets from SAM or crystal. They all works very well. So I don't know, like, what's a big difference, but actually they are all perform good for the quantification.
Yeah, in a word, very similar.
Yeah, very similar. About one thing is for the SAM tools, they just put so many functions, so it has become more and more complex. So the crystal is a little bit simpler than SAM.
I have a question about the Callisto and Salmon versions. So whenever there is a release of new version, does the Galaxy update to get updates with the new versions of Callisto or Salmon?
I just forgot to mention. So in the Galaxy interface, let's see for the Salmon. And then the SAM quant. So here on the top, you have versions. So you can select different versions of the quantification.
Okay.
Yeah.
Okay, thank you.
I think we are slightly behind the schedule. I think we should take a 10-minute break.
Yes, so for the second part, I'm going to talk about the 3D RNA-seq app for the differential gene expression and differential alternative splicing analysis. So let's begin with a question. So for biologists to perform RNA-seq data analysis, how long does it take? It's 1 week, 1 month, or 1 year? So I asked my supervisor, who is a biologist, the same question, and he gave the answer. It's impossible. Then I told him, if you use my 3D app, it's possible. So I'm going to give the answer in the end of the presentation. So for the 3D RNA-seq app, what is 3D? 3D is not the 3 dimension. It refers to use RNA-seq data sets for differential gene expression, DE, differential what's going on. Yeah. Yes, refer to the, I just point the later. So it's referred to use RNA-seq datasets for differential gene expression, DE, differential transcripts usage, DTU, and differential alternative splicing DUS analysis. The 3D app also can be used for the isoform switch analysis. So the isoform switch is where the transcripts isoforms see reverse relative abundance. For example, here, the two transcripts isoforms just reverse their abundance. So here I'm going to show the expression profile of a gene from the study of Lepidopsis in response to the code. So for the data sets, we have 26 time points, and then the Lepidopsis will first place at 20 degree, then translate to 4 degree, and then 4 degree day 4 of acclimation. So in the plot, the top line is a gene level expression, and the other lines are the transcript expression. So from the plots, we can see both the gene and the transcript, they have a significant expression change across these 10 points. Here is another example. The top line is the gene expression. It doesn't have a very significant change across the 10 points, but the transcripts isoforms, they have very significant change. For some of them, they even form a isoform switch. So the expression change are very complex. To study this complex change, it relies on bioinformaticians with highly special skills. They often take months to get the results, and then there's big variations in the results because the quality of the results, because different people, they use the different methods. Then the biologists, they get totally frustrated. So what's the solution for these issues? The solution is a 3D RNA-seq app. The 3D RNA-seq app is a very easy to use tool because it's web-based graphical user interface designed for biologists. It's all inclusive. It's not only for differential gene expression analysis, it's also for differential alternative splicing analysis. It can be applied to plants, animals, and human studies. It's very accurate because it uses state-of-the-art methods and it has good controls of false positives. It's very flexible for complex experimental design, like the pairwise as 10 series and the development series datasets. It's also very rapid. So the 3D app is a cutting-edge analysis pipeline because it includes many different steps. For the first step, you take the quantification result from the salmon or crystal. Why we choose salmon or crystal? Because they are sustainably faster and they require less memory and disk usage, and they are very accurate. Then the TX import our package, take the quantification result and generate expression matrix for the analysis. So the TX import our package, it can correct for the potential changes that due to the library or the transcript runs biases across examples. Then we also have various steps for the data preprocessing. For example, an important step is to remove the low expressed genes and transcripts. But usually to remove the low expression, it's very difficult to determine a cutoff. So we use a mean variance trend plot to tell users the optimal parameters for the filters. Because the RNA-seq reads, they are assumed to be negative binomial distribution. So these mass equations showing the relation between variance and mean. So the mean variances, they follow a monotonically decreasing trend. For example, in this formula, we can see when the mean increase, the variance should be decreased. So in this plot, we show the mean variance trend. The x-axis is a mean and the y-axis is variance. So this is from the real RNA-seq datasets. So we can see at the low mean value part, so it's low expression part, we have a drop trend in this end. So we need to remove this part. Then after remove that, we can see the drop end has been removed. So the whole trend of the mean variance is follow monotonically decreasing. So in this way, we can select change the parameters, increase a little bit, and to see if the trend, the drop end has been removed. If it's not, we can change the parameter for the filters, and until we get a pattern like this. So this is a way to tell users the optimal parameters to use for the filter. Then we also need to remove batch effect. So what is batch effect? Batch effect is caused by biological replicates being prepared in different lab conditions. For example, the different biological replicates were taken in different time or different locations or by different people. So we can use a PCA plot to visualize the main variance of the data sets, and then the examples of the biological replicates, they should, if we see some batch effect, then the examples of that biological replicates, they stay in a separate cluster. For example, here is a PCA plot. It's our Arabidopsis data set. We have 3 biological replicates, and then the biological replicate 1 was performed in 2010. So we can see they stay in a different clusters to the other 2 because they are performing to different years. So after we remove the batch effect, we can see the biological replicates, the 3 biological replicates, they stay in the same clusters. So this is how we use a PCA plot to identify the batch effect, and then we can remove them. And then also we need to do data normalization. Before data normalization, we can see the recount, the distribution across examples, they are very different. Then after normalization, we can see they are in the same, they are in very similar distributions with very similar mean and similar variance. And then for the 3D analysis, we use a Riemann-Wurm-Arc package. So Riemann-Wurm-Arc package is a very popular tool. It has been used for RNA-seq data analysis for many years. So how we do the expression comparison with this package? So here I just show some pictures. For example, for the first picture here, we can see the top line gene, and we have 2 transcripts. So they all showing very big expression change. But for the transcripts, their percentage or their proportions to the gene levels, they don't have the change. So we say these transcripts, they are DE only transcripts, and the transcript 2 is also DE only transcripts, and the gene is DE only genes. So in the second plot, we can see the gene level, it doesn't have big change. And then for the transcripts, they have big expression change. Also, their proportion, their percentage to the gene level also have big change. So these 2 transcripts, they are DE genes and DTU transcripts. And for the genes, because it doesn't have differential expression change, so it's only DAS genes because it can have some alternative splicing change. Then in the third case, we have the transcripts. We can see it has no abundance change, but it has some percentage change. So it's a DTU only transcripts. And then for the other transcripts, the DEE, DTU transcripts, and for the gene is DE and DAS genes because this gene has this expression change, also has alternative splicing change. So by analyzing the expression and the percentage, or we call it percent spliced change, in this way, we do the 3D analysis. Then to showing the result, we use different plots and different figures. For example, here we list the numbers of DE genes and DAS genes, and then we use box plots to showing the up-down regressions, the Venn diagram, the volcano plot, the goal annotation plot, and the heat map. Also, we can do isoform switch. And then in the end, we will generate a report, include all the methods, all the pictures, and all the parameters you used for the analysis. So in a summary, that means all good things just come in a small package. Then back to the question, how long does it take for the whole analysis? So the answer is 1 to 2 hours for beginners. So if you don't believe it, I'm going to do a demo of the 3D RNA-seq analysis. So the data sets I'm going to use is a lepidopsis in response to cold data sets. So I pick 2 samples from the day 0 and to the same time points from day 1 and the same time points from day 4 for the demo. And then we can because they are in different days and different temperature, then we can compare the expression change between them. And then the data set has 3 biological replicates and 3 sequencing replicates. Then with these data sets and the reference transcriptome ATRTD to quasi, we can do Salmon quantification. We can use a Galaxy GUI, or we can use a command line. And then the quantification will be imported to the 3D app for the analysis. So for the 3D RNA-seq analysis, it requires 3 inputs. The first input is a metadata table. So it's showing the information of the samples, and then we need list the conditions, and then the sequencing replicates labels and the biological replicate labels. So here I'm going to use a Galaxy quantification results for the analysis. So I need list the file names from the Galaxy quantification. So if we open these files, it's showing the name of the transcripts and the runs and then TPM and the number of reads aligned to the transcripts. Also, we need another input, the transcript gene association table, because we are going to do the analysis both at gene level and transcript level. So we need to know their relationship, like these transcripts is belong to this gene and these 2 transcripts belong to another gene. So, yeah, so I'm going to do the demo. And so here is so here is how the input data set look like. We have this metadata table showing the sample information, and then we have this mapping table showing the transcript gene association. So they are all in CA3 format. Then we have this quantification results. So here we have the quantification from Salmon Galaxy. So you can see all these are the outputs from the Salmon quantification. So in the 3D RNA-seq app, it looks like this. You have different page. For example, the introduction part, and then the data generation part, and data preprocessing, the 3D analysis, the time series trends and function plot, either form switch, and then in the end, you can save all the results and generate a report. So in all these panels, you have different boxes to import data and set parameters. So also, if you don't know how to do the analysis or prepare the data sets, you can always click these question marks. So the help information will showing up to tell you how to do the preparations and how to set the parameters. Like here, and then like this. So to do the analysis, we need to start from the data generation part. Then for the first step, we select the sample metadata table. So here we have a metadata table, and then we need to select the transcription association table. And then you can also visualize these tables here. So you have uploaded these tables. And then in the next part, we need, so So the quantification, we need to select them because we need to upload them at once. So here we have set the quantification files. We need to select the set one. So it take a while to get upload. So there's a warning message showing up here. Please double check whether the quantification files are saved or you select the correct quantification method because it's from Galaxy, it's not from command line. So here I need select from Galaxy. Then it's done. So in the second step, I need to select the experimental design. For example, here we want to study the different time points. Or we want to study different time points at different days. So for the data sets, we have sequencing replicates. So we need to select the biological replicates, also the sequencing replicates labels. Then we need, it's from the quantification of Galaxy. So we need to select the columns that showing the name of the quantification and add the information to the table. So this is the table. Then the next step, we need to generate the expression matrix by the TXT import package. So we can select different methods. Though the length scale TPM is recommended because it's using the average transcript length of examples, also the library size for the to balance the expression co-examples. So we just click run. It will take a while to generate the matrix, and then it's done. Then we can move to next step for data processing. So we use different figures and plots to tell users the optimal parameters for filters. For example, for the low expression filters, we use a CPM. CPM is count per million reads as a cutoff. And then we need cut and set up parameters for how many examples we need to cut. So then we do filter and make the mean variance trend plot. So we can see here is the plot before filtering and the plot after filtering. We can see here still a drop and here. So we could increase the number of samples to cut. And then the drop trend has been removed a little bit. If it's still there, we just increase the cutoff again or cutoff again. So this is very hard to tell you the optimal parameters to use. So now we can see the drop trend has been removed. Also, we can see the gene levels. So drop trend has been removed. So it's okay. Then we can move to next step. So here we can also plot the number of transcript per gene. So it's just a plot showing you the number of multi-isoform genes. So these plots here for each plot, because you can make changes. So you have to save this plot. Also, you can save this plot. And then for the next step, we need to use a PCA plot to visualize the batch effect. Here we click the button plot. So we can see for biological replicate 1, examples stay in separate clusters. So once we see this batch effect, it means we need to remove it. So in the next step is the made batch effect. Data has batch effect, yes. But here you should pay attention that the batch effect is you must have a kind of reasonable explanations why you will have this batch effect. Like in our case, this batch effect caused by the biological replicates when the samples were sampled at different years. So you have explanation for that. So we can remove them. So you remove the batch effect. And then you take a while to do the analysis. and then you update the PCA plot. So you can see after the move batch effect, the examples is they stay in this very similar clusters. So we can also save this plot. Then we can visualize the saved plot. So if you are not happy with this empty space here, you can change the parameters and to save. For example, we change the x and x limit for that. Then we save the plot again, and then we visualize it. So this plot looks much nicer. So then we can also save this plot and visualize the saved plot. So after the move batch effect, it looks like this. So by using these parameters to control your plot, it can help you to generate a publication quality figures. Then the next step, we need to do data normalization. We click this button, then we can see before normalization, the distribution is different across these samples. So after normalization, the distribution has become more similar. So after the data pre-processing step, we can go to the next step for the 3D analysis. So for the 3D analysis, this table showing the experimental design, the different conditions you can compare. So in the second step, we need the contrast groups. So contrast group is a way to compare these conditions. So for example, here we want to compare the 10.2 of day 1 to 10.2 of day 0. And then we can add another line to compare 10.2 of day 4 to 10.2. So we can set these kind of contrast groups to do the comparisons. And then we can also compare the mean of two groups to the mean of other two groups, and then we can compare the mean of day 4 to the mean of day 0. Or we can also do another comparison, the difference. So this is, for example, we want to compare the difference of these two groups, and the difference of these two groups. And also here, so we generate a contrast group. So here you can see tables. We have many cases for the comparison. We have these individual conditions compare. We have these group mean compare. We have these group difference comparisons. So then with these contrast groups, we do the 3D analysis. So in the 3D analysis panels, as I said, we're doing expression comparison according to these pictures, and then we can set different parameters like the p-value adjustment method and the way to summarize the dust string levels. Then we do different cutoff for the adjusted p-values, the log4 change. Then delta p is so delta percentage of splice change. Then we click the button. Then it will take a while do the analysis, so it's done. Then we have different ways to visualize the result. For example, the tables of the testing statistics, like DE genes and dust genes, and then we can also make the profile plot in this panel. So we just need to type a gene name, and then we make a plot. So you will get a plot like this. Also, you can divide the plots according to different days and then make the plot again. So it has been divided into different days, and then you can save the plot. Then you can also have the percentage splice change plot. So for example, here we use different colors to showing the percentage of the transcripts. So for example, these transcripts .1, the percentage is very small here, but it becomes very big in day 1 and become very big in day 4. So it has very significant percentage change across the different days. Then we we can visualize a saved plot as look like this. You can also change the dimension, like the plot height, and we save it again. So your saved plot will look like this. And then we have the tables to show in the number of DE genes, DAS genes, DE transcripts, DTU transcripts in different contrast groups. We have this Venn diagram to showing the difference and the consensus parts of different contrast groups, and then we can save the results. Also, we have this up-down regression plot. So we can save the results like in different contrast groups, all these number of up-down regression change. So when we save it, we can its plot look like this. So if you are not happy with these X labels, you can load it, and then you can save it again. Then you have a plot, the labels has been located. So if you're not happy with the color of these plots, you can also change the colors. For example, we change it to blue, we change it to red, then we save again, and then visualize the plot, you will have like this. Then we can also do the volcano plot. So the volcano plots look like this. And then you have the DE DAS comparisons. You have DE only genes, you have DE and DAS genes, you have DAS only genes. Then you also put the DE DTO transcripts. And then you have a picture showing the unit set of all these results. So you have expressed genes and the DE genes not expressed, and then the DAS only genes, and then you can also have the transcript level results, the DE, DTO transcripts. Then you have the Venn diagram. It's another format to showing the DE, DAS result, showing the comparison of DE, DTU result. And then you can also save this plot. So the next page is for the time series trend analysis. So in sometimes we do the RNA-seq experiments. We do like our Lepidopsis data sets. We have different days, like you have different group, and then in these groups you have a number of time points being measured. So we can compare the trend of these groups. So this is the purpose of this trend analysis. So to proceed the analysis, yes. So this show in the panel for the analysis. So we have the 10 points in different day group. So he's showing that we have this day group, we have these 10 points, and then we want to compare this trend of different days. And then we want to set a contrast group to compare the trend. So generate contrast group. So statistically, it compares the time trend to see if the trend in 3 days, they are the same or not all equal. So the same as the 3D analysis, we can set different parameters, and then we just click the button, run it. So we have the result. For example, for the DE trend, we have this gene, the contrast group, and the adjusted p-value for that. And then we have the numbers of the DE time series trend or DAS time series trend number, and the DE transcripts and DTU transcripts of the trend. So we can also make a plot for the trend here. So, and then we divide it by time, the group of day. So we can see the trend of these 3 blocks are very different. Then in the next step, we can make heat map for the results. So the heat map for the DE genes or for the DAS genes, for the DE transcripts or DTU transcripts. So for example, we make a plot. Then you can see this is a plot of the DAS genes. So we have a heat map. Also, we can have heat map for the differential express trend analysis, like the trend of differential alternative spliced. So you have the time series dust trend genes in contrast group. Then for the clustering method, you can choose different methods for the clustering, and you can cluster the genes into different groups. Then you can also save the results. Then the next step was for goal annotation analysis. So the 3D app can take in the goal annotation results and then make the plot. So for example, here I have prepared the goal annotation result for the DE genes. So I use another software to generate the goal terms, like the molecular function, the biological process, and cellular component. I have this goal term information. I have different statistics of the significance of the goal term. So in this panel, we can upload this table. So the DE gene goal annotation. So we can upload these tables, then we can choose different statistics and then make a plot. So the plot has look like this, the goal annotation plot. So if you don't know how to prepare the inputs for that, you can click the question mark, so it's showing different steps, or you can use a gene list to generate the goal annotation tables that import to the 3D to make a plot. Then, so here is for the isoform switch analysis. So the isoform switch, we're doing the analysis to compare between two contrast group, how many of the transcripts they are reverse, they are relative abundance. So if you want to go to more details, if you want to get more details, you just go to a publication. The TSI is the TSI is the publications here. So you can go to that paper for more details of the method and the parameters for that. So here in this panel, we can choose time series analysis, or we can compare pairwise isoform switch between different conditions. So then we only need click the button, and then you're doing the analysis. So it will take a while for that. And then if it's done, we can set different parameters to filter the isoform switch. So for example, we do the filters. So this table showing the results of a pair of isoform and their switch statistics. Then we can also make a plot for the number of switch between different contrast groups. For example, day 1, 10.2, day 0, 10.2, they have about 100 switch. So we can also make a switch plot here. So we type the name of a pair of isoform and then make a plot. Then we can see we have a switch here. Then we can save the results. Then in the final step, we can save all the analysis results. And then we can generate a report. And then we can click the download button to download the results. Yeah, so we just download the results. So once everything done, I have the downloaded results here. And then if I unzip it and click in, so you will get different folders. So you have the data folders. So here I just save all the intermediate data sets in R.data format. So if you are users, you can go to these files to get the intermediate data sets. So we also have these figure files. So it contains all the figures you saved. And then we can go to the results folders. So it has saved all the testing statistics, the number of genes and transcripts, and then the gene and transcript list in the heat map. All these results you can find here. And then for the reports, you go three format reports: the Word format, the HTML format, and then you can also convert it to PDF format. So if we open one report, we can see that it's so it has master section, it has resource sections, and then it also has the supplementary information part. So in the master, it's lists all the master for this data preprocessing, for the 3D analysis, and then the contrast groups used there, and the parameters, the cutoffs, all here. And then in the result part, all the pictures you saved has been inserted to the report. So you have tables of the numbers, you have different pictures. And then also in the supplementary material part, I list all the file names. For example, in the figure folder, I list the name of the figures and give it a bit description. And then the files in the results, and also you have the description of the files. And then for the intermediate data set in the R format, if you are R users, you can access these different elements in the data list to get the information of the intermediate data sets. You also have the reference. So so here's also I prepare some tips for the 3D analysis. So for the first one is you should understand your experimental design properly and provide proper labels in the metadata tables. For example, in our experiments, we have different time points in different day. And for this condition, we have different biological replicates. We can label them as biological rep1, rep2, rep3. But so this is correct labels. But if you label as rep1, 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 rep2, 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 so this is wrong label. And if you label as 1, 2, 3, 4, 5, it's also a wrong label. So you need to have a proper understanding of your experimental design and then provide the proper labels for the metadata table. So also for the quantification informations, the format of command line and the Galaxy output are slightly different. So here I'm showing you how it looks like. So for example, if it's a Salmon Galaxy, you will get the file name files with an extension of tabular. But if you get a quantification from Salmon command line, you have an individual folder, and these folders will have a quant.sa file. So if you open these files, it's just the same information, the name, the range, the TPM, and the number of reads, but it's just a different format. So the same for Crystal. So for the Crystal Galaxy, you will get the tabular output. So if it's a command line, you will have the quantification in individual folder, and in each folder you will get abundance TSV. So the abundance TSV is a table showing the quantification. So in the metadata table, if you're from the Galaxy, you need to provide the full name of the quantification. So if it's from command line, you need to provide the information of the folder that contains that quantification result. So if you want to go to more details, you can go to the Galaxy user menu. In the end, you will get how to prepare the information in the metadata table. Then another important tip is how to prepare the transcript gene association table. So there are two cases. The first case, the gene name is a substring of the transcript name. For example, the barley and the rice and the Arabidopsis. For example, if the Arabidopsis one, the gene name is just a subset of the transcript name. So in this case, we can go to the salmon quantification or the crystal quantification result, because the first column, we have the transcript names. So what we can do is we take the transcripts from here and then use some tools like Excel to cut the transcript name and get the gene name. And an important note is the transcript names in the transcript gene association table must match to the transcript names in the quantification. So another case is a gene name is not a substring of the transcript name. So for example, the mouse dataset or the human, so the transcript name and the gene name, they don't have configuration between them. So what we can do is you can extract the information from two files. The first one is the GTF annotation of the transcript. So we open a GTF file, it looks like this. So in the GTF, in the description columns, we have the gene ID, we have the transcript ID, so you can extract the information from here. So, or we can, the other way we can do is we can extract the information from the description line of the transcript sequence FASTA file. For example, here we open it, we have the sequence of the transcripts, but we also have a description line, start with the bigger than sign from here. So the name after the bigger than sign is the transcripts, and also we have a gene name here. But the problem for the FASTA sequence files, usually the maybe the description line is not complete. So maybe you only have the transcript's name, you don't have the gene name. So it's not always work for the second case. So also in the 3D app, let's go to the 3D app. In the data generation part, so we recommended you to provide the CF3 format, the gene transcript association tables, because it's the file is small, but also you can import the GTF, then it will automatically extract the information from the GTF. And then you can also provide the FASTA and then it will extract the information from there. But the problem is maybe it not always work, because it depends on if the descriptions in these GTF or the FASTA files, they are complete or not complete. So if it's not complete, probably you can't upload them here to get the transcript gene association table. Then there's another point is, do I need to remove the batch effect or the outliers? So the batch effect is examples of the biological replicate staying in separate circles, like the case we showing before. So you have these separate clusters for one replicate. Then we need to remove batch effect. That batch effect has been removed, so you will have these consensus clusters. And then the other cases, not all the biological samples of the biological replicates is in the separate clusters. Only like one or two samples which look like outliers. So in this case, you don't need to remove the batch effect because it's not a batch effect. So what you can do is you use the room weight method in the 3D analysis to reduce the effects from the outlier. So we go to the 3D app, and then in the 3D analysis part, for the step 3 3D analysis, so here you can select the room weight method, and then you do the same analysis, and then just click the run button, and then you will reduce the effect of the outlier. Yes, so we only get about 20 minutes left, so I'm going to show you two applications of 3D app very quickly. So the first one is for the time series of lepidopsis in response to code. The second one is the effects of the dexamethasone on mouse brain tissue. So the dataset is the same dataset we, as Renshan mentioned, for the lepidopsis in response to the code. And then we do the salmon quantification and 3D analysis. So we have so many different time points in day 0, day 1, and day 4. So we set contrast groups to compare the time points from day 1 to the same time points to day 0. Also, we can set contrast groups to compare the same time points of day 4 to the time points at 20 degrees. And then we do DE gene and dust gene analysis, and then we do the cutoffs for the p-value and the log24 change, and then we summarize the DE dust gene result. So here is a 3D result. So we have the express genes, we have the DE genes, and then we have not DE genes, and then we have this DE only, and both DE, DE dust, and then dust only. So the important thing is for the only AS regulation, so the dust only genes, we have about 1,700 genes. So they only showing significant change at transcript level, but they don't have change in the gene levels. So these parts of genes never been studied by other people because they often just throw them away. So also we use a heat map to visualize the expression. Then we can see the identified DE genes. They show very good patterns of this dynamic change between different time points and different day. Then we also compared the 3D results to currently known code response genes. So we have about 3,000 novel code gene response genes. About 42% of them are dust-only genes. And then, so here is just showing some numbers. So that means our 3D pipelines is actually very good to do that. So we can also study isoform switch because we found once the plants will transit to the code, the transcript isoforms, they reverse their abundance. So we just do the isoform switch analysis and then make a plot of the numbers of isoforms between different time points. So we can see the majority of the switch just occur within 6 hours after code. And then these isoforms, a big proportion of them, they involved isoform switch. Involved protein coding isoforms. So the second application is the effects of the dexamethasone on mouse brain tissues. So we take the datasets from a publication, it's from 2018. So the datasets, it has the tissues from different brain regions for male and female and has different treatment. So in the original paper, they use the HR for the analysis. Then they only do the analysis for DE genes and then identify any fault change or adjust p-value cutoff for the they didn't have any fault change cutoff, or they don't do FDR controls for the p-values. So I just analysis their datasets and then do the comparisons. So in our analysis, we will do very stringent filters for the fold change, and also we do adjustment for the p-values. So we can see, we have, although we do a very stringent analysis, we still have about 5,000 new DE genes being discovered. And then the data set is also being analyzed in another paper by using method sleuth. So the sleuth identified about 3,200 DE genes, and then for the 3D analysis, it identified about 3,700 DE genes. So we identify more genes. Then we also identify the dust genes. So in our pipeline, we can resolve the DE genes and dust genes. So here, just make a Venn diagram to compare the 3D results and sleuth results. Also, we identify the dust genes, and then. So what does this resolved DE and dust gene mean? So here I make the goal annotation plot. So for the sleuth DE genes, and then in their plot, in their results, I do the goal annotation. I found the terms related to DE and the terms related to splicing, they just all mixed up and they didn't separate. So in our study, we have these DE genes, we have dust genes, we have separate this information well. So that's the advantage of our 3D pipeline. It's not only for DE gene analysis, but it's also for dust gene analysis at alternative splicing level. But because the 3D pipeline is the contrast group settings were flexible, so we can do the very complex comparisons. For example, we can compare the sex-specific DE and dust genes. I'm not going to describe the details of the result. And then also we can do brain region-specific DE and dust genes, and then you can use different ways to show the results. And so just to make a summary, so for the impact of the 3D app, so it enables the biologists to analyze their own data sets very quickly and very easily. So it's a huge save of cost and time. So it's a real game changer in research planning, because for the whole analysis of RNA-seq data sets from data generation and for the 3D analysis, probably it's only take 3 to 4 months. So that means in a general grant period, probably you can design a lot of experiments and get a lot of results. So it also unlocks the discovery potential of the public available data sets. So the 3D app won the Best Innovation Award from the School of Life Science at University of Dundee. Then currently the app has over 3,600 users and about 880 returning users from 55 countries. That's being applied to various plant species and also animal and human studies. So if you want to get more details, please go to our publications. And then just acknowledgement. Thanks again for the SEFARI, for the fundings for the workshop, and also the workshop organizers. And then for the 3D app development support, I get funding for my postdoc and the supervision from Renshan, John, and Robbie. And I asked RNA-seq experiment support from Nicoleta and Christian, and then the technique support from Gordon Stephen and Ian Mile. And then all those people who test and the app and give feedback. So that's it.
Brilliant. Any questions from? So again, for the demo there, I think there will be a handout with step-by-step guidance for running through 3D RNA-seq as well.
Yeah.
And what Wenbin showed there is actually, I'm not sure if you guys realize that it's actually a live demo. So that data was uploaded to that website server. and it was analyzed in real time and downloaded. So if you analyze the data, you will see exactly the same of what's going to happen. So I think yeah, go ahead and try it, because there has been a lot of people using the software, and we got feedback from people who never analyzed sorry, any data before, and they're able to go through that 3D RNA-seq pipeline. So what I mean is don't have that kind of mental kind of inhibition that this task is so difficult. Once you go through it once, next time it will be just a few minutes for you to run through again, to change the parameters, adjusting things, etc. Sorry, Carmen, you got a question?
Yes, because I don't have the data. Has been some delays, so we don't have data or something. If you could recommend any repository that I could maybe download data, or for Barley or something, where I can find data for practicing, because we don't know when the data will be available.
So if you want, we actually have some Barley data available for previous trainings. Is that in the tutorial? Probably not. So we can probably send that training data to you.
Yeah.
As well for Barley. If you look particularly for Barley, I think in previous trainings we have made three sets of data available, which is a Arabidopsis, one set of Arabidopsis data, one set of Barley data, and there's another set of mouse data.
Yeah.
Is that true? Yeah.
Yeah.
So yeah, if we could probably, is that data still online?
I need double check for that.
Yeah, so if you want to get familiar with the pipeline, just get any set of data would be sufficient.
Yes, but I don't know where to go to take the data. Sorry.
All right, okay. So if you I'll share my screen. Okay. So can you see my screen right now? So you should have this 3D RNA-seq app manual sent together, set to you guys already. So you can see that's a step-by-step instructions there. So if you go down input files, oh yeah, example data is here.
Yeah.
So this is.
Can you click a link and get the example datasets? So I think it's a 3-set datasets available there. Let's just double check.
Okay. Yeah. Okay. Let's have a look.
And then click in. Oh, it's empty.
Oh, what happened? All right. Okay. No, I think this is only one set of data.
Okay.
This is a Arabidopsis data.
Yeah, so I will provide another. So if you go to the.
So yeah, so for the Arabidopsis data, you can just download from this Dropbox and follow this workflow from here. So you basically have each of the steps when we will explain what each part of that web page is, how you set parameters, etc. And more importantly, I think that YouTube video really helps in terms of guiding you through this. Yeah, so it's actually step by step. Just try that, because when we developed this manual and this app, we actually tried with undergraduates. Just give them the data and give them the manual without explaining anything and ask them to walk through it. So they actually managed. So don't worry too much. Just give it a try. If you do get stuck, we are always available for help. Good. Any other questions or concerns? Or maybe advice how we can make this virtual training better?
Could I ask a question, please?
Yeah.
Yes, go ahead.
So going right back to the beginning with the data, is it necessary to quality filter the raw RNA-seq data before you put them into Callisto and Salmon, or does it actually make negligible difference? It's something I've never really understood. So the data that you're taking right at the beginning, has that already been quality filtered to a certain Q threshold, or?
Yeah, we did do some quality filtering, but I think the mainly reason is that we use the same data for transcriptome construction as well. But I don't think necessarily you need to filter your data before running Callisto or Salmon. But you should bear in mind, you should look at the mapping reads coming from the result. If there's some issues with your data, you can see the mapping reads from Salmon or Callisto outputs will be very low.
Right.
Yeah, so you can see those reads actually they couldn't find a place for the read in the transcriptome at all. Or for example, a large proportion of your reads actually doesn't map to a particular transcriptome. So that should be some red lights you should think about.
Yeah.
Yeah, but would those just then, that's what I mean, maybe it's negligible because you're actually then going to take, if you set a minimum cutoff for mapping, you're not going to use those. The anomalous reads or the reads that are not that aren't genuine. You won't use those in the later analysis.
Yeah, of course you won't use that. But the issue is, so for example, I can give you one example. I was analyzing a small RNA data set for potato. But then when I, in the process of my analysis, I realized the mapping rates are very low. So usually I expect, for example, 70% of mapping. But for that set of data, they only got 10%. So I come back to them and mention this issue to them. And then they later tell me that the protocol failed. Probably they didn't capture any of the potato small RNAs. Right. So I think that's basically the type of issues that you should be aware of. Although you can continue with analysis, but the reads left for you to making the statistical inference may be not enough anymore. So, yeah, that would be something that I would be aware of. So doing the quality check is always recommended.
Right. Thanks.
Yeah.
Hi, can I ask you a question?
Yeah. Go ahead, Rodney.
Yeah, I mean, do you have any idea what is the publication timeline for BART version 2?
We would think in 2 or 3 months' time. But if you need early access, talk to us. No problem at all. Particularly, you just email Robbie.
Yeah, true.
He's PI on that project. So I think.
Yeah. Okay. Thank you. I'll get in touch with him.
Good. And I think we deliberately leaving this 1 week between the lectures and the one-to-ones is to allow you some time for you to explore.
Yeah.
When you come back with more questions, so that you can, make the best of the 3D RNA-seq and make it work for you. Sorry, there's a chat. Is there further questions? All right. So in the chat, when we already send the link for the 3 different data sets, so you could probably
Oh. Just see. Mention we lost you a while.
okay. Hello?
Yeah, we can hear you now.
Okay. Yeah, I was just saying that when we have sent a link for 3 testing data sets for 3D RNA-seq, you can explore anything. Just take any data set you're interested in and have a go.
Yeah.
Okay. Any suggestions as how we can make the training service better for you guys? Or what do you want to do from now? From here? Just have a go.
Great.
Okay. Yeah, that sounds good. That sounds reasonable to me as well. It's a strange without this intermediate feedback. Yeah. It doesn't allow us to. Doesn't allow you guys to run it immediately.
So so my GitHub page include all these links. So if you miss some links of applications, you just go to GitHub page. You can search GitHub 3D RNA-seq, then you will find my GitHub page. Then all the information is there.
Yeah, so if no more comments or questions, I think we will probably call it close. Yeah. And thank you very much for everybody participating in this virtual workshop.